Here we're looking at biochemical evidence, and we find that almost all living organisms use the same biochemical material. We all use DNA, all of us use ATP as our source of energy, chemical energy. Why do all living organisms use these same materials? Those two work. Same amino acids we use mostly, and there are some variations in amino acids. And the same genetic triplet code is used for all life. So there's a biochemical connection for all living things on the planet. And as we'll see later on today in lab, there are even other signs of continuation of life, like for a script that gets written over and over again. And sometimes some of the old material is left behind even though it has no more use. We have the same developmental Oct genes. These are genes that are responsible for determining where limbs go, how our body develops, what patterns are set. People who have experimented with these genes in animals like mice and flies and other organisms have found that if you mess with these genes, you get limbs or they don't normally appear. Extra limbs. They're really important, and we all share some holy talents. So recognizing this, one of the things that became pretty obvious to scientists once we had developed biotechnology, DNA technology, the ability to sequence DNA to actually look at the very genes of all life, was that instead of looking at how things look on the outside, we could start looking more closely at molecular evidence in terms of how closely related organisms are. This chart here says the number of amino acid differences compared to human cytochrome C. Now, cytochrome C is a protein that plays an important role in the electron transport chain. We didn't get a chance to look into that in detail, but we know this is an important process by which a lot of ATP is generated. And cytochrome C is a key protein in this process. So much so that any organism that is generating ATP through an electron transport chain is going to have it. So when you look at it and you compare other organisms' cytochrome C to the human cytochrome C, this is some of the stuff you get. For example, chimpanzees, I think, is identical. But if you look at monkeys more distantly, they have two amino acids that are different. Now, all the amino acids that make up cytochrome C, and we're talking hundreds, only two differ between a human and a monkey. So that tells us that monkeys are probably fairly closely related to us in terms of the evolutionary tree, in terms of where our common ancestor was found. If you look at pigs, there are a total of 9 amino acid differences between their cytochrome C and ours. Ducks, 11, turtles, 18, fish, 20, sauce, 30, yeast, 51 out of 100. Yeast look a heck of a lot different than us. I mean, heck, most of you probably agree that a fish looks a lot different from us, and yet they're only, the difference is only 20 amino acids. But it's interesting that not only that this evidence supports what we see visually. People look at monkeys, they look at humans, and say, oh, yeah, they have 4 limbs, they have grasping limbs, they have 4 facing eyes. It seems pretty obvious that they're more closely related to us than pigs. We walk around with no clothes with a snout, and then they're going to be more closely related to us than ducks. But not only does it show us in terms of how they look, but if you actually look at the molecular evidence, if you look at something that has nothing to do with how they look, how they generate energy, we find that that is borne out, that same sort of visual relationship is also supported in terms of the molecular evidence. That's, again, biochemical evidence for an evolutionary relationship. So greater variation equals more evolutionary distance between the organisms. And I'm going to show you another diagram or chart that will emphasize time and evolutionary distance too. Now, before I do that, let's talk about how humans are classified. We are eukaryotic. All of our cells have nuclei, They all have membrane-bound organelles. Part of it's called a supergroup, and this is a relatively new taxonomic division that you don't really hear a lot about. Obviously, still content. And what they did is start creating these supergroups because they wanted to start grouping up the Protista, this very group of eukaryotic organisms that are mostly single celled. They wanted to understand their relationship to the multicellular organisms, because multicellular organisms derive ultimately from single cell ancestors at some point in time. And so we're in the same group as the group of Protistas that use flagella. And you think, well, where do we have flagella? Well, we only have one sperm. That's the remnant flagella that we still have the genetic code for. We only use it for one cell type now. But presumably at some point ancestrally there was a cell that was this way of getting around in the world. But now humans just use it for one. There are Protistas that still use flagella just to get around, swim from one place to another, make their food, to move. Then we get into kingdom, which is sort of the older taxonomic nomenclature. It used to be the highest level of organization for all the eukaryotes, but then they came up with domains. So, we call it super group. So the old way, the kingdom Animalia, was a group that we're part of because we're multicellular. We're motile, we can move about, and we are heterotrophic. That means we get our energy by consuming chemicals. We don't stand out in the sun and photosynthesize for energy. We can photosynthesize some vitamin D, but that's about the extent of our photosynthesis capabilities. We rely upon taking our energy in by eating it. It's interesting because there are members of the kingdom Animalia who are not motile once they hit their adult form. They are motile when they're in their larval stage. So generally being the kingdom Animalia, you have to have some state where you have some ability to move. That may be challenged, though, ultimately be through genetic evidence again, but right now I think it stands pretty tight. So underneath that category, there are a number of different phylum, right? And one of the phylum includes basically chordata, and there's the non-chordates that we're not part of. So in order to be part of the chordata group, you are going to have notochord, which is something that we developed during our embryological stage and hopefully gives rise to our vertebrae. And pharyngeal pouches is something we share with all the other members of the phylum chordata. Under that classification, then you have under phylum, you have different classes. So there's the class that includes birds, for example, which also nowadays a lot of taxonomists want to put the dinosaurs in with the birds. There's some debate about that. So there's class Aves, which includes birds, and then our class Mammalia. And we're different from birds, so we don't have feathers, and birds don't have hair. We have hair, we have mammary glands, birds don't have that either. Below that, getting even more specifically within the different groups, there are different groups of mammals, and including the placental groups and non-placental groups. But even more specifically, we're part of the order of primates. That means we have a large brain, we have forward-facing eyes, we have grasping hands, and we have nails instead of claws, flat nails. There's some primates that are called a grooming nail, which is actually pretty claw-like, or misspoken, it's pretty claws. Family, our family is Hominidae. All hominids are believed to have an upright stance, and that's how taxonomists treat that. There's really not a whole lot of genetic evidence that they can chase after on this one because most of our hominid relatives died out a long time ago. We're the last of once great clan of Hominidae. Great because it's larger, great because we all have cheese. Our genus is Homo. Basically means the ability to make and use tools, and our species is sapiens, so we are Homo sapiens. Homo sapiens, unlike all the other homos that we're going to talk about, known to have speech centers, and maybe even more importantly, known to have the ability to generate art. This creative ability is so critical to our species because it's believed to be what has driven all of our scientific advancements, our ability to be So this is often applied to all living things in terms of different classifications. I can at least be for short the domain, kingdom, phylum, class, order, family, genus, species. We just have a little mnemonic to help us remember that. Darn, king, goat, good, often, fine, good, sauerkraut. So those all start with the same letters. Darn, domain, king, kingdom, Philip, phylum, good, class, often, order, fine, family, good, genus, sauerkraut, species. So further building upon that, looking maybe at where we fit kind of in the giant tree of life, particularly at the level of domain. So this is where all three domains are being portrayed here. You have all the eukaryote, all the cells that have nuclei, and then these cells don't have nuclei, and they're often considered to be fairly closely related. The archaea and the bacteria, these are often called archaebacteria. But doing some genetic analysis again, this time instead of looking at the cytochrome gene, because actually there are organisms that don't engage in electron transport, so they don't have cytochrome at all. So you can't make that comparison. So they turn to something that you do have to have, and that is ribosomes. All living things have ribosomes, because ribosomes make proteins. Bacterial ribosomes are actually considerably different than our ribosomes. They look very similar. They have a large subunit, a small subunit, but they're different enough that there are certain antibiotics that affect them that don't harm our cells. And so we use those to treat disease, wipe out bacteria that are making us sick. Nevertheless, they do have ribosomes, so you can look at their ribosomal genes. And if you compare the ribosomes of the eukaryote group and the archaea group, you find that they're actually more closely related than the archaea are the bacteria. In fact, there's some idea that the archaea were probably the cells that kind of engulf some bacteria to create mitochondria, and they went on to become multicellular or become proteins, and then beyond that became multicellular. So these may very well represent some of the ancestors. Well, it's interesting because these organisms live in some of the most extreme environments. They live down in Yellowstone National Park. They live deep in the sea near volcanic vents. So they're pretty way out there in terms of really live in some pretty hostile environments. Maybe they actually represent some of the first forms of life. But bacteria have been around for a very long time. It's hard to say. Further evidence suggested that the fungi, now when I went to school and was learning my degree in biology, you had to take a sequence which included zoology, which is focusing on the study of animals, and then you took botany, focus on the study of plants. But often, but always, in my case too, we study the fungi at the same time, because at the time, people thought fungi were more closely related to plants, because fungi just didn't move, neither did plants. Animals moved. Now fungi must be more closely related to plants. When they started looking at the ribosomal genes, they found that fungi are more closely related to animals. And the reason, well, not necessarily the reason, but the other factor that people were considering is feeding styles. Plants photosynthesize, whereas we eat other organisms to get our energy, all the animals do, and that's what fungi do too. So their mode of life in terms of how they acquire energy is very progressive, very much like ours. So maybe it shouldn't have been so surprising when they're focused too much on this non-motility and ability to move. But actually, there are types of fungi that have some motion. So again, maybe that was a false illusion. If we think about humans and we look at all the apes that currently exist on this planet, and we think about who is our common ancestor, ribosomal genes tell us that our last common ancestor with apes is 7 million years ago. So an ancestor from 7 million years ago gave rise to all of the apes. Now, that's not all the primates, that's just looking at apes, so that includes chimpanzees, gorillas, and orangutans. But it doesn't include old world review of monkeys. You have to go farther back than that if you want to find a common ancestor for all of those. And again, hopefully we get a chance to explore that a little bit more go to the lab today. You can also look at mitochondrial DNA if the organisms have mitochondria. This, of course, is absolutely useless when you're looking at bacteria that don't have mitochondria. They are what made mitochondria. The the thing about mitochondrial DNA is if you look at it, you're actually able to trace more recent changes in evolution. And so they use mitochondrial DNA to look at where humans evolved from. They look at changes in populations across this globe, and so they find the oldest shared examples are present in humans in Africa. So they basically have come to the conclusion that we evolved in Africa. We have since migrated out of Africa. That's covered our planet. Your mitochondrial DNA is something that you inherit from only from your mother, so it's also used to actually look at people's lineage in terms of their maternal lineage, in terms of who their mother was, who their mother's mother was, and so on and so forth. So it's useful in that regard too. So I had quite a surprise. I actually had my mitochondrial DNA looked at, and it turns out that my ancestors that came from Genoa, Italy. I mean, I love Italian food, but I had no idea what it was. Otherwise, I would have Italian in me. But somewhere my mother had some ancestors from there. Probably Romans, would be my guess. The Romans got everywhere. All right, so we are primates. We have these forward-facing eyes. We have grasping hands. Primates are generally divided into two groups. Now, nowadays, this has been thrown in a little bit into chaos because of genetic analysis. One group that they thought, one of these groups, subgroups that belong in one of these bigger groups, turns out may not belong there at all, and that's the tarsiers. It's actually looking at ribosomal, RNA, and other factors. You find that tarsiers are actually more closely related to apes than they are to either orcid and lemurs. So the tarsiers up here at the top, the big round eyes, bush monkeys, I haven't heard of just seen one of these in Africa. They're pretty cool. And then lorises and then lemurs. Genetically, these two belong in the same evolutionary phylogenetic group. But this is what they call a paraphylogenetic group. They basically group all together, maybe because of their eyes. I don't know. Their eyes actually, the tarsiers are about the size of their brain. And then the anthropoids include the monkeys and the apes and humans. So we have African apes here, we have Asian apes, we don't have any monkeys. So we're going to look at some monkeys eventually here, the white-handed gibbon and the orangutan or the Asian apes. These are more closely related to us than those up there. Primates have mobile forelimbs. This one allows us to get around the trees, and hind limbs, and with the exception of us, most of them have opposable big toes. Whereas we don't, we've lost that ability. We all have binocular vision that allows us to judge distance, and it's believed that originates from the need to be able to judge the distance between one branch and another, because we come from tree-dwelling ancestors. So somewhere along the road, humans gave up the tree life and hit the ground and started running. And we can't binocular vision because it's awfully useful to be able to judge distance, particularly when you're trying to throw a spear at prey. So we probably can't have one binocular vision because it's useful in terms of our hunting ability. We have a large, complex, and highly folded brain. So even those big-eyed tarsiers are pretty smart little animals because they've got a pretty well-developed brain. We have a reduced reproductive rate compared to a lot of organisms because we engender a lot of parental care and we get involved in that. There are fish that lay eggs, fertilize them, and then off they swim. And, good luck to all the children. They make a lot of eggs, they make a lot of children, so that enough survive to replace the population, the parents at the very least. Otherwise, think about it, they're always increasing, and we'd be covered in fish, and we're not. So there's a balance there in terms of how much gets reproduced and how much doesn't make it. On the other hand, we invest a lot of our time and energy in taking care of our children because we don't produce that many. We need them all to survive if they're going to reproduce and replace the current existing population. We've been so successful at this that our population has continued to grow over the last several centuries, and now it's presenting us with some challenges in terms of human population. And get further into the details of classifying us if we look at how we compare to chimpanzees, our closest living relative. They also have closer living, they've either been subsumed, hybridized with us, or died off. We'll talk about some of those here in a moment. But chimpanzees are still alive, yet they're obviously not us. They share about 98% of our DNA. It's identical. Our spine exits from the center of our skull, where a chimpanzee's spine exits more from the rear end of the skull. The shape of our spine is S-shaped, and this is a direct result of our ability to stand upright. It was what had to happen. We'll get a chance to explore that also. Whereas the chimpanzees, although they can go upright for periods of time, they generally also spend a lot of time going around on all four limbs. That's because of the really long arms. You look at the relative length of arms of humans versus chimpanzees. The chimpanzees' arms are long enough to reach the ground comfortably for them, not to mention that their actual phalanges are developed in a way that allows them to do knuckle walking on the ground. So they have just what they call an anterior curvature of their spine. There is a little bit of a forward curvature to the neck because they both are kind of up. And they do it regularly enough that they have a curvature there. That's interesting, I'll mention that. Looking at the pelvis, the human pelvis tends to be more shaped like a bowl. This is also necessary to help something that stands upright, not only to walk, but also to give birth, where a chimpanzee is more elongated and more narrow, more focused on tree climbing. So that's better for them. Our femurs are angled inward. And this is a key characteristic for organisms that walk on 2 feet. In terms of mammals, we're the ones that have been adopted and invested entirely in this life. Others are still walking around on all 4, whereas a chimpanzee flow outwards. And you can see that if you've ever seen a chimpanzee walk. So they're really not built to walk upright. So they don't do it for very long. They do it to display mostly to show anger or dominance, as the case may be. And we have this little thing called an arch in our foot. Really between the arch in our foot and this curvature of our spine both create a spring for us. This is a spring in our feet that allows us to bounce down our feet, move along. This is our spine acting as an S-shaped spring so that we can also, our torso can absorb that shock as we trundle along with our 2 legs. Chimpanzees don't have that. They have flat feet. You may have heard people have flat feet, but usually it's not as flat as the chimpanzee foot. And typically, there's been a lot of cases where people have done reverse flat feet simply by reducing the amount of cushion they have in their shoe wear and taking it right. Any questions? So, an evolutionary tree is a working hypothesis of the history of life. This is a very specific one looking at mammals. So, the mammalian ancestor enters the trees, maybe they were always there. And what this is also allows us to see that connection that I was talking about earlier. We see the lemurs down here. We look at the tarsiers. Tarsiers shared a common ancestor with monkeys and apes a little bit less than 61 years ago. Versus if we look at lemurs and tarsiers, their last ancestor was shared more than 60 million years ago. So that means that they're more closely related to monkeys and lemurs. We can say that we shared an ancestor with this group before we shared an ancestor with this group, what they did. So that gives you a relatively time period in those 2 groups. And they determine this using what's called a molecular clock. If you have DNA that is not being selected for by natural selection forces aren't acting upon that DNA in a way, then mutations will be acquired by the DNA at a fairly regular rate. The frequency by which mutations occur is based on statistics and random chance. Over enough time, those mutations are guaranteed to happen. So they accumulate at a steady rate, and you can use those genes to kind of look at how long ago different organisms evolved from a common ancestor. So by looking at those molecular clock, we're looking at those accumulations and mutations, that's what allows them to say, oh, well, parasites started at common ancestor over 60 million years ago. And there is some debate on what's the best molecular clock, what looking at DNA and genes, you have to kind of do some research to figure out what their accumulation rate is, because there is some variance there based on the sequence of nucleotides, how many of the G's versus A of the G's and C versus A of the G's are present there. So we know that there are two groups of monkeys, the old world monkeys, the ones that live in Asia and Africa. They have a nose that protrudes. Think of a baboon as an extreme example of this. They have no prehensile tail. Some don't even have a tail at all, but if they do have a tail, they can't use that tail to grasp limbs. That's what prehensile is. New World monkeys are found in South America, well, I have this in Central America. I should know this because I went to Panama and we fed some monkeys there. My friend ate little squirrel monkeys. They have a flat nose and they have a prehensile tail. They can use the tail to grab limbs, so that effectively gets the fifth limb from getting around in the tree. And they're quite afraid of doing this. And then we have the hominids, which is basically us plus chimpanzees, gorillas, and orangutans. So we can give it something to fit into that category. You can see the given are the outlier group here. They share their last ancestor more than 15 million years ago, where orangutans, gorillas, and chimpanzees are closer to 12 million years ago. And obviously, humans and chimpanzees are most closely related, and then next is gorillas. So the homininians are referring more directly to us and our ancestors. And basically we agreed that what we share with all of those groups are the bipedal. We walk around on two legs. And for a long time there's a lot of debate about whether are we the tool users, and they found chimpanzees using and making their own tools, although they tend not to pass them down from generation to generation and make them eat them and then discard them when they're done. So it's not quite the same as when humans would actually make rock tools and literally pass them down from generation to generation and teach how to make those tools. Although chimpanzees do teach too. But then once we kind of made it kind of murky, and so the whole tool using thing went out, and then people thought, well, maybe it's the brain, their brain was made that special. And so all hominids should have a certain size brain to fit into that group. And then they found evidence where most of the hominids have a bigger brain even than chimpanzees. But really the defining feature that kept popping up was basically walking around with two legs. That was it. Our brain kind of developed after we freed our hands so that we could use our hands to be doing other things like making tools. And then those who were smarter at making tools got more food, produced more, so the brain kept growing. Maybe that also has to do with a better diet. Still a lot of debate about it. Archipithecus, which I think we'll see in a movie today, walked upright but had opposable toes. So it was kind of weird. There's a ape-like ancestor. They think, again, there's some people who say, no, this is a extinct group and has nothing to do with our lineage. But they walked upright, but they had the ability to climb in trees very effectively. And they lived in a forested landscape. So that also kind of drew a lot of hypotheses to the wind, who thought that there was a change in the climate and the forest kind of died had to learn to stand upright, see above the grasslands, to see potential predators and avoid them. And now, though, it seems like this, and more recent evidence has shown that organisms learn to walk upright even in the trees. So there was an advantage to being able to walk upright in the trees. And I'm thinking maybe looking more at chimpanzees because they have some ability to stand upright. You can sort of see how there might be a progression from that to a more regular walking upright all the time. At least one hypothesis that I heard suggested that it was important to be able to reach branches higher up by standing up and grasping onto them. It's a little weak because they can take care of a lot of that, but maybe as you got bigger, so the bigger body plan that became more important. Whatever it was, one reason or another, walking upright became advantageous and it stuck around, and then eventually it became important because we left the trees. And there's still some debate as to when that happened. Maybe walking upright became an important feature just to leave the trees on a temporary basis and be able to take advantage of life on the ground and then still go climb right back up. The next member of our family, at least pretty much everyone agrees on, Ardipithecus is still debated quite hotly sometimes, but the Australopithecines are pretty much recognized as the ancestor of all the groups that became Homo this, Homo erectus, Homo heidelbergensis, Homo habilis. There's a number of them. These organisms had an upper body that was very ape-like, but their lower body was completely bipedal. They didn't have an opposable thumb anymore. So they had definitely given up living in the trees and were now on the ground. They weren't that tall. They were maybe 3-4 feet height at the most. Particularly tall, but somehow they were able to survive on the ground. A lot of very large and hostile predators, bigger than the predators that we have today. We're talking about millions of years ago. So then they started thinking, all right, so we have these Australopithecines, and then we have these other ancestors referring to show more and more similarities to humans, and that's when they decided that we call these groups Homo. They decided to use Homo to them versus the Australopithecus. Although frankly, Australopithecus africanus is very Homo-like to me versus Homo habilis here, which is a little bit more primitive with those big old cheeky bones there. So rather than looking at that, what they focused on is their brain size. They decided that if your brain size is 600 cc's, cubic centimeters or more, and if you had human jaws, teeth, and used tools, that you could fit into Homo. It seems sort of arbitrary to me, but since we can't get DNA from fossils that are 1 million years old, it's kind of all we got to do after all. Chimpanzees have a brain about 400 cubic centimeters, and they're quite intelligent. They can learn sign language. Some of them, not all of it, but they can certainly learn and quite smart. So 600 cc's was a boost up. Not as big as our brain, but still pretty large. Size isn't everything, as they say. Homo habilis. There's Homo habilis here. This is Homo habilis, but I think it doesn't look like human. But Homo habilis lived almost 2.5 million years ago to 1.5 million years ago. So that was a good run. That's like 1 million years. It's been around. His brain size was 775 cc's, so it's definitely running some brain power compared to 600 cc's. Looking on the inside of the skull, they have some pretty good skull fossils for Homo habilis. They see areas where we expect to see the speech centers, referring to speech area versus motor speech area. And that leaves imprints upon the skull. And so there's a suggestion that those areas might be enlarged. So there are questions remain, were these organisms, were these early humans culturally inclined? Did they have cooperative hunting? The jury is still out with that one. But they did find evidence for their tools. Tools were scraping meat. The tools, however, were relatively crude stone tools. Nothing more than hand active. A hand active does not have a handle, just the rock. Regardless, the rest of the handle is the axe. But they're definitely tools, and they're made by striking other rocks to create a sharp edge. Homo erectus came onto the scene a little less than 2 million years ago, so they actually have curve along a lot of the time curves would coincide with Homo habilis there. So we have to wonder if there was some interbreeding here. Sadly, they don't have a skeletal Homo erectus here, but it's definitely more human than that. I describe it as a very successful species, and I postulate maybe there's more than one species because they've been around for a long time. We're talking almost 2 million years here. There's fossils that suggest it was around as long as early as 40,000 years ago, which is a long time to our brain right now. But in terms of fossil evidence, that's a blip. That's very short. It probably was around the same time as us, very shortly, and certainly Homo neanderthalis. And it's pretty much assumed that Neanderthals and sapiens ultimately derived from erectus, even if phylogenesis might have been in between fossil type that led to Neanderthals and sapiens. It's a fossil mess because we have a few fossils, and these populations probably numbered in the millions, and so it's really hard to get a good feel for the representation of that population of so few fossils. But what evidence we do have, Homo erectus had 1,000 cc's of brain power, still short of ours, but very respectable. Males are big, up to 6 feet, so we're getting more human size. A lot of these earlier homos were 4 foot, maybe 5 foot maximum. Females tend to be smaller. There's a lot of sexual dimorphism, so they describe that as sexual dimorphism. The evidence of looking at their hips and their leg bones suggests that these were organisms they could run. They could actually fully get into all the strike gait, so they were probably doing successful cooperative hunting. They used fire, they established camps. And this was the first group that got out of Africa as far as we know. All the evidence that we have, they left Africa, they got as far as Indonesia, maybe even Polynesia. So, a subgroup of this Homo erectus called Floriensis that was actually went the other way. They became smaller. They weren't as tall. But they're one of the groups that are actually around up to 40,000 years ago. And they lived on an island, pretty much isolated from us. And maybe that's what affected the world. This is sort of this is more looking at, I think, how ideas can be postulated and then discarded, in case you can call it. This is just created in an instant and stick with it. Or trying to describe to you how hypotheses have changed over time based on evidence. And that's the crucial thing about science, is that if you've got the evidence, you can make a case for your hypothesis, then you enter into debate. You can be part of the scientific community. There's an openness to it in terms of looking at evidence and considering it in an honest fashion. So for a little while, there was a couple of different type of hypotheses involving where humans evolved. So one of these suggested that since Homo erectus was so widespread that it didn't make it to the New World, wasn't in South America or North America, but it managed to spread all through these areas, all the way down to Indonesia, all the way to China. That maybe that Homo sapiens had evolved more than once, that there was sort of just a given result to get a lot of time to pass by, and Homo erectus gave rise to a version of Homo sapiens, and then that different populations of Homo sapiens appeared. They called that the multi-regional continuity hypothesis. And then the replacement hypothesis more focuses on this idea that we originated in Africa, and then we migrated out and we replaced all these other populations, and we hybridized with some of the other ancient hominid lines, and that gave rise to Homo sapiens today. But that, basically, the bottom line is that all the evidence points to that we evolved in Africa and then moved out into the wide world. But there is also definite evidence that we did interbreed with some of the ancient groups that were currently existing out in that wide world, including Neanderthals. Neanderthals was massive 1400 cc brain. They were done. They had a very protruding face. You can see us, Homo sapiens, on the left here, and Neanderthals on the right here. And massive brows for the orbits of their eyes. So if you look at their forehead region, that sticks way out. But there's definitely evidence that they interbreed with humans because we've been able to get DNA out of Neanderthal fossils. And we've been able to sequence it, and we've been able to look for those sequences in existing modern human populations. We found that some individuals have as much as 4% of their DNA came from Neanderthals. So somewhere along the line, Neanderthals and Homo sapiens interbred. Neanderthals were much more robust, more muscular, so it made them a very physically imposing creature. They evidenced some culture because there's evidence that they buried their dead with flowers and ceremonies. Cro-Magnon is basically an early version of Homo sapiens, and the book states that they didn't interbreed with Neanderthals. I'm not clear on the evidence on that, but we're not going to worry about it too much. There's another group of ancient humans that are kind of a mystery group. They're called the Denisovans. I looked at the evidence that's available, and it seems to me that Denisovans are, at least genetically, are very, very similar to Neanderthals. A lot of the pictures that I've seen, the thing what makes them a mystery is we don't really have any good complete fossils. We've got some pieces of fossils, and they've been able to extract DNA from them because they're not that old, 4,000 years ago again. They lived in what's now today Russia and Asia. Some scientists consider it basically as just the Russian Asian version of Neanderthal because they aren't different enough Neanderthal based on genetic sequence. And their DNA is found in our populations. Again, there's Denisovan DNA in our population. So it'd be really nice to find a good fossil of this particular group, but I suspect they're going to turn out to be just sort of a subgroup of Neanderthals with similar features. You know, it looks like Cro-Magnon has a smaller brain. Again, it's not just the size of the brain that matters, that's how it's organized, how many folds it has, and then the depth of those folds. What really set modern humans apart so far, at least, is we haven't really found evidence that Neanderthals had the ability to do this, is art. And mostly, particularly true when it came to wall paintings. All the wall paintings that we've ever found are attributed to Homo sapiens. Meaning that Neanderthals never did wall painting. Maybe we just don't have any evidence for it. They did, someone erased it, or whatever the case may be. Even if they do ever find evidence that maybe Neanderthals did, they'll probably start saying, well, they were mimicking us. Let's try to hold on to art. They're trying to figure out why modern humans are still here and Neanderthals aren't. What gave us the edge? And it's believed to be our creativity. Our tools were a lot more sophisticated than Neanderthals. Neanderthals have pretty sophisticated tools, but Cro-Magnon came with some amazingly delicate arrow head points and other fish hooks and other tools that just gave them an extra edge in terms of getting food and reproductive advantage. Anyway, that's what I got for human evolution. Why don't we take a 10-minute break and then we'll dive into some human skeletons. Had some knee replacement surgery there. And then there's one tibia, and the end of the femur there. My future right there. Maybe I'll review this if you're lucky enough. I'll review. So, what is bone made of? The bone's made of bone. Well, it's not so simple. It turns out bone has a lot of different tissues. Obviously, one of the number one tissues we should think about is bone tissue. We've already talked about tissues in this class. We are going to do a lab devoted to tissues too. It's going to be a little bit late, but it sort of fell out of the schedule. So we'll have a chance to review all of these different tissue types we're talking about. We're going to look at some detailed structure of bone tissue in this chapter. So fear not if you're like bone tissue, what's that? I'll tell you this, it's hard, got a lot of calcium in it, like some not the only thing we find, though. We find some cartilage tissue. The only lights that work in this room right now. I've drawn a few words for us up there. That's a great protein up there. And maybe a little tibial tuberosity there. That's a little too much tibial tuberosity. That's okay. And we'll draw a nice trochlea here. And then a capitulum. Another lateral epicondyle. We'll keep our way back in like that. And on the surface of the head, and on the surface of the capitulum and the trochlea, is a substance known as cartilage. Specifically, it's called something called hyaline cartilage. It's often called smooth cartilage. It's the most common cartilage in the body. It's found on the end of all of your bones. Where bones come together and articulate, you find this type of cartilage because it's smooth. We want to reduce friction where bones meet because friction equals pain for us. If this cartilage wears out, which it does for many individuals, then they start doing knee replacement surgeries and take other measures. They have some left that's starting to wear out, you start to feel that in that. Maybe change your diet habit, take some chondroitin sulfate or glucosamine to try to ebb the tide there. Dense fibrous connective tissue. I don't think I can do justice to that, so I'll point at the screen. They're showing that they peeled away. I can make a copy of that. They peeled away part of the tissue there. And they're showing us like lots of fibers. And this is a membrane called periosteum. And it goes all the way around the entire bone. All the tendons, the ligaments that tie into it are holding on to it. So it helps it encapsulate the bone. It makes it a true organ by having this capsule around it. This protective layer thing. And why would bone need that? Well, bone can be fairly brittle. This makes it a little less brittle. It gives it some cushion to it. So it's a dense, fibrous connective tissue. Go on to take anatomy, we'll talk about it. It'll be a dense, irregular connective tissue. That's okay. Dense fibrous will work. And there's nerves in here. Right? If you break a bone, it hurts. If you haven't broken a bone, well, good. No, it hurts like that. There's a lot of nerves in your bone. You break it, they equal send signals to your brain, and you'll be yelling. That's not fun. I don't know how well you can see it. Turn the lights down again. If you can come on, that would be awesome. Yeah. 2 cents, right? Yeah, I'd love to see that. Well, I was just more connective tissue. Then we have smooth muscle. We have all 4 tissue types. There are 4 major tissue types in the body. So actually, on the outside of the blood vessels, in the big blood vessels, there are going to be muscle fibers here. And they will also have their own nuclei. Smooth muscle, not cardiac muscle, not skeletal muscle. They call it organ muscle. So that's found in bone too, so all of the different tissue types. Now, that's kind of physically part of the bone, either inside, outside, it's incorporated in the bone. This is considered outside of the bone, it's considered structures that lie outside of the bone. Again, where do you draw borders? It's hard to say. So they don't have I did not include a picture showing any good ligaments here. Do I have a good picture? what I can do is I can draw my infamous knee joint. I had a knee joint up there earlier. I'll just do that. I don't want to get rid of this. We'll draw a femur. And femurs tend to be rounded at the end. That's a terrible femur. I'll draw the tibia. So this here is the patella. Get some bones in already. And then coming off the patella, coming out here to the patella. Here are the quadriceps muscle. Which means that this is the quadriceps tendon. And then going down here, into the tibia, is the patellar ligament. What I can tell you is that ligaments connect bone to bone. And tendons connect muscle to bone. Ligaments connect bone, tendons connect muscle to bone. This is the same darn stuff. With quadriceps tendons and patella ligament, it's made of the same tissue type. It's a dense, regular connective tissue. But because this part is connecting this quadriceps muscle to the patella, it's considered a tendon. And this is connecting the patella, which is a bone, to bone. This is considered a ligament. But ultimately, the quadriceps muscle pulls on this and is able to, you're able to kick your leg out because of it. Because of that connection. And there are other ligaments in here. There's what they call the cruciate ligament, the anterior cruciate. You don't need to know this. I'm just showing you that there are other ligaments here. And then there's a posterior cruciate ligament, and then the fibular collateral ligaments. All kinds of ligaments are holding these two bones together, so they don't spread apart. So ligaments are made of dense parts of connective tissue. So are tendons, and they either help hold the bones together, become ligaments, or they help hold the muscle to the bone, which is what produces movement. So the muscles contract, and that results in the forces pulling on the bone, and it causes the bone to move. So our ability to walk, run, hit, talk, all of that due to those ligaments and tendons. Questions? Functions of the skeletal system. What does the skeletal system do? It supports all our soft tissue. We have a bunch of soft tissue that is held in place by our skeletal system. Holds us up. Next soft tissue. Literally, it's considered classified as a support connective tissue. This is kind of its primary job. All right, movement. That's better. Muscles pull on bone. Provide leverage. We didn't have bone, we just sort of squiggle around on the floor. We didn't get very far. Even snakes have bones to move around. The bones give support and structure to their muscles so they can produce that motion allows them to squiggle across the floor more efficiently. Protection. Your skull protects your brain, ribs, protects your heart, lungs, your blood vessels. I gave you a list, but I don't want you to remember that. I just want you to focus on protection and give a couple of examples. Production. What the heck does it produce? What does bone produce? Do you have any ideas? Red blood cells. Excellent. We have red bone marrow that's constantly making blood cells. If you break your hip, one of the things they're worried about is your blood cell production. You're becoming anemic because you don't produce enough blood cells. Storage. Fat, calcium, minerals. Storage is so important. Calcium is so important. We think about calcium, we think about bones, we have to get enough calcium in our diet to make sure we have nice strong bones. And yet our body sometimes defeats us anyway, pulls calcium out of our bones, and gives us osteoporosis. If we don't get enough calcium in our diet, it's not so much bones that our body worries about right away. It's worried about our heart. It's worried about our nerves. And our nerves and our heart rely upon calcium ions to function properly. If you don't have enough calcium in your blood, if you don't have enough calcium stored away in your cells, those cells stop working and you die. Your body is going to prioritize that function over your bone structure first, because if you're alive, well, you can try to deal with rickety bones. But if you're dead, then well, it doesn't matter anymore, right? So it will scavenge out of your bones calcium in order to make sure everything continues to work properly. If you don't want to have weak bones, make sure you get enough vitamin D and calcium in your diet. You get vitamin D by getting out there in the sunshine, or you can take a pill, or you can drink your milk because they put vitamin D in your blood. Which is convenient because what else is in milk? Calcium. So you take milk, you get your vitamin D, you get your calcium. And on top of all of that, though, you have to do a third thing to really give you the best chance of maintaining your bones throughout your entire lifetime. You know what that is? Magnesium. Magnesium is good. That's not it. Exercise, resistance training. If you don't stress your bones get lazy, they get weak. They adapt to whatever lifestyle you adapt to. So, anyway. Storage, right? Magnesium too. That's what I mean. Yummy. Some nice yellow marrow in there. The bones are filled with marrow. That's the fat in there inside that medullary cavity. That can be right in the center of the bone here. The intronic fat rolls there. You get a knife, you get some of that, you spread on some toast. So good. Well, unless you're vegetarian, but dogs love that stuff. People do too. Oh, here's an activity for you. Take out a piece of paper separate from your notes. Put down your name. Today's date is the 4th. Okay, it's my dad's birthday. Equal blow up and body blow up your I would like to point out that even the little pinky finger here, the fingers in there are all sort of long. They all have these features. Obviously, the humerus, which is what they're portraying here, is a perfect example of a long bone. But one of the things that they have is they have two epiphyses. Two epiphysi. And the ends of the bone you find spongy bone. I think I got a good job that they've done at the top of that bone to show a cutaway. And the spongy bone looks like a sponge, except for it's hard as bone. And then this cartilage, although it shouldn't be called hyaline cartilage, it's never enough names in anatomy, also known as articular cartilage. And that's in recognition of its function. Its job is to be articulating with the next bone. In this case, that would be the scapula that would be not bloody evidence of bone there. Again, that's hyaline cartilage, like cartilage that it is. And then this centerpiece here, this is called the diaphysis. And that's the D, or the inside the diaphysis is this medullary cavity, this internal cavity. So we can draw a cutaway view of that. This internal cavity here is the medullary cavity. That's partly filled with yellow marrow. I showed you that picture of the toads. This yellow marrow is fat. So we have lots and lots of fat reserves inside of our bones. It's a great energy reserve for us, allows us to be active continuously throughout the day, even more so than the light age of liver. In between the epiphysis and the diaphysis is this region known as the metaphysis. I don't know why they never really draw it there, but basically in this region there is this line, in a mature bone it's a line called the epiphyseal line, but in the immature bone, a bone that's still growing in length, as a child, it's still getting taller, is an epiphyseal plate. So it should be cartilage. Hyaline cartilage. Epiphyseal plates and growing teeth, children, made of hyaline cartilage. I mentioned the most common cartilage in the body. And ultimately, when you're done growing, it becomes replaced with bone. And then there's this remnant structure called the epiphyseal line. It's more of a scar. Adults, bone, no growth. Scar-like. Remnant structure where the plate used to be. I mentioned there's this outer membrane on the bone called periosteum, made of dense fibrous connective tissue. And this is contiguous with the ligaments and tendons. So if you were to look at the surface of these bones, you would also find some periosteum there. patellar ligament here ties right into it. So by doing so, it's wrapping around the entire bone. So the patellar ligament looks like it's attached in one place, and it is. You can't pull a ligament out, and it will not detach the entire periosseum. But that's the idea, is that it's getting support around the entire bone. So it's continuous with ligaments and tendons, they attach directly into it, and it gives support around the entire bone. Additionally to that, on the inside of the medullary cavity is another vertebrae. And this membrane is called the endosteum. So peri means around. So it's around the bone. And endo means inside. Osteum refers to bone. Osteum refers to bone. So the endosteum is the membrane on the inside of the bone. This is completely cellular, by the way. It's not connected tissue. There are no tendons or ligaments that tie into it. It's just a reservoir for bone cells to produce new bone. Okay, now we get small. Microscopic structure of bone. Unpack them. If we consider our bone model over there, and if we were to take a slice out of this bone and you were to turn it sideways, it wouldn't be perfectly round. There would be this medullary cavity in here, right? If we were to take a slice of that bone out and we were to expand it many, many times, then we might see something like this. We see some spongy bone on the inside lining here. We see these circular structures, some of which are not complete. Now we find that one of these circular structures has a name, basically. All of these circular structures are considered the functional unit of bone. They're called an osteum. And if you were to take that osteon and you were to flow that up even further, you'd find that it has some structure to it. This part here is called the central canal. This is the blood supply. You can see they've drawn up one of the osteons here. They've pulled out the rings, and in the center you see red and blue. Those are the blood vessels along through here, the central canal. You've got the red and blue, the arteries and the veins. Imagine bone is very vascular. There's blood vessels not just in the medullary cavity, but in the bone itself. Every osteon found here. Within between these rings that we see, there are these little chambers. Planets around the sun. Except they're oval shaped. And living within those chambers are cells. We've got a little nuclei there. So the chamber is all lacunae. That's basically an old Latin word for room. The cell is present inside the lacunae, so we'll say lacunae with and osteocyte. So osteo again means bone, and CYTE means cell, so it literally means bone cell. Ranging out from the lacunae are these little channels. By the way, you have to imagine that this is a thin slice of bone. So there's actually a lot more osteocytes here at different levels. And so when you look at actual real slices of bone like this, you see all these channels throughout these veins all over the place. And these little channels are called canalicula. Little canals. This allows one osteocyte to communicate with another osteocyte so they can coordinate maintenance and repair of the bone if they need to. It ultimately guarantees their supply to the central canal. And that's where they get their blood supply, that's where they get their nutrients, that's how they get rid of their waste. So this intercommunication is critical to how the bone functions. And these functional units of osteomes are what make up compact bone. So compact bone refers not to the spongy bone that we saw earlier, or this stuff here, but the really dense stuff that forms the wall of the bone. The real thick outer wall of the bone is made of compact bone, and it runs up and down the length of the bone on either side. Look on my graph showing an osteocyte there and inside of this. So spongy bone looks like this. I found this image, which is a pretty good drawing. This is a real head of a fever here that cut away through the compact bone, the outer surface of the bone. So we can see the red marrow on the inside. It's really hard to see it. There's like a little, it's very spongy, there's a little yellow marrow center in the center there. This is just a better drawing to show the nature of spongy bone in all these spaces. So you don't find osteomes in spongy bone because the blood vessels, this is where we're making blood cells, so there's plenty of blood here, plenty of blood supply, plenty of oxygen and nutrients. But we do have a name for these struts, these spicules, and that is trabeculae. So those represent the struts of the bone. A good word, strut. I've heard spicules, struts. You guys have another name for them? What do they look like to you? Beams? No idea. No. We'll go with me then. That basically gives a lot of support in this region, where there's a lot of stresses coming from different directions. Whenever you have a joint where two bones meet, there's tendons and ligaments that pulling in all these different directions. You want lots of stress also oriented in different directions so that the bone can withstand those forces. Compact bone is great for compression. When you actually are compressing it, it's very resistant to it. Spongy bone not so well. But for forces pulling on bone, you want spongy bone. I mentioned there's a lot of red bone marrow here. It's right over the head of the femur there. So that's where we produce red blood cells. So red bone marrow. red blood cell production. Questions? All right, let's take another 10-minute break then. So cartilage is very similar to bone, and then houses many. And the difference is the cells that are inside the cartilage are called modular cells. Right, chondromic cartilage, and CYCD building cells. Cartilage doesn't have any blood vessels though. Still, if you think about the fact that the articular cartilage was relatively thin on this bone, any nearby blood vessels could potentially supply them with their nutrients they need. But it does tend to slow down the healing. Cartilage, if you break it, it doesn't heal as quickly. And it's particularly true as you get older, as most things are. So I mentioned hyaline cartilage is known as smooth cartilage. It's the weakest of the three types of cartilage. Fibrocartilage is found in a few places. Remember, hyaline cartilage is the most common cartilage, but you can find fibrocartilage in between the vertebrae. Sideways view of some vertebrae here. In between the bodies of the vertebrae, there will be a little piece of cartilage. And then the fibro. This is an intervertebral disc. It is the strongest cartilage. Elastic cartilage, if you take the ear and bend it down like so, and let it go, it drops back up. That's because you have elastic cartilage in the ears because of that elastic recoil. Lying over your voice box here, or a structure called the glottis, is this little piece of plaque called the epiglottis. So when you swallow, the epiglottis closes on top of the glottis so that food and drink doesn't go down into your larynx and you don't choke. So when you're done swallowing, it's going back up so you can breathe out again and talk, breathe. So the epiglottis is made of elastic cartilage also, so it can go down and spring back up when you're done swallowing. ears and the epiglottis are really the only two examples of loss. Any questions? Okay, so the entire skeleton, all of your bones are divided into various different categories. The two broadest categories are the axial skeleton and the appendicular skeleton. We're going to talk about the axial skeleton first. Bones that protect vital organs and serve as the center. So in this image on the screen is the bones that have sort of a bluish cast to them. That is the axial skeleton. So the axial skeleton would include Well, you tell me, what do you see up there? What's in the axial skeleton? Skull. Exactly. Anybody else? The ribs. No, we can't include the pelvic girdle. No, that's not ridiculous. Good. Any other ideas? What about the sternum? And the vertebrae? Or even you can say vertebral column. That's good. Everything makes the center. So we'll start with this one here. Let's call the cranium. In particular, because the cranium is referring to the part of the skull that forms the cranial cavity where your brain is. All the thinking is going on. So these are a list of all the bones that surround your brain. But before I start down that road, I want to talk about this skull right here. That is a baby skull. Tragic thing. At the very top there's a little soft spot. That's what we call it, soft spot. Right about here. And actually, babies have more than one. They have one here, they have one back here, they have actually a total of 6. They have one there, one over here, and one over there. This is the biggest one up here. It's one that most people will notice on a baby's head. The fancy name for that is a fontanelle. And it forms because basically the baby's skull is not completely turned to bone. All of our skulls, everyone's skull starts off as a membrane. The flat bones of the cranium start with membranes, and then they slowly get replaced with bone during fetal development, with the exception of this spot here, this spot back here, and these 4 spots on the sides. That gives the skull of the infant flexibility, so when it passes through the birth canal, it can not get stuck unless it pushes its way through. Also adds additional protection in babies in case they get dropped, they fall over. They tend to close at about 16 months, but then until then there's these soft spots that are relatively weak points also between the brain and the outside world, so you have to be careful. Actually, you can see on the front of that skull, there's like a little fissure going forward, and you don't have that fully in the joint all this way. Completely fused to form one single big frontal bone that forms our forehead. It also is protecting the frontal lobes of the brain. Now we're going to see this word a few times, sinus. Okay, so sinus. Here's my renditions of metatarsal bone. The little suture here. Right about here, there is a cavity, and this is called a sinus. Specifically, it's a type of sinus called a paranasal. It means it's near the nasal cavity, and it's connected to the nasal cavity. It's got mucous membranes in it. It produces mucus that you're into your nasal cavity, blow your nose. It influences your voice production, but also is believed to lighten the skull, so the skull's not too heavy. But I think it's there also because of the need to produce excess mucus, because we have to keep the nasal cavity clear of infectious agents. And that excess mucus is kind of critical to that. On either side of your cranial bulb are two big flat bones that have a slight curve to them called the parietal bones up here. The walls of the cavity, so this would be a bone that would continue right here. Do that more smoothly. What bone is that? Well, the parietal bone, there's two of them, one for either side. So that's the exception here. Most of the cranial bones are singular. There's two there that are paired up. There's not a lot of features on the parietal bone, so one thing I'm going to ask you to do is whenever I mention a feature in my outline for one of these bones, I'm expecting you to remember it or basically go to that on your exam aid page when you're creating that for the lecture. Okay? The occipital bone, there's only one. This is the bone back here. This is the back of this bone right here. The protuberance here called the external occipital protuberance ligament and mid-shaped edges on the flexural patella. Your husband went through all the work. That's right here. But the feature I want you to remember is this foramen magnum. Foramen means hole. Magnum means big. It's a big hole. Why do we need a big hole like that in our skull? We think it has to pass through there. It's fine. Arteries, blood vessels. Actually, surprisingly enough, part of your brain passes through here. Or the brain stem. It's the lowest part of your brain. In fact, the lowest part of the brain stem, the pons and the midbrain are protected well inside the cranial cavity. The medulla oblongata skips out here. And part of the medulla oblongata is the cardiovascular centers that regulate your heart rate and blood pressure. And you can't live without that. So there's a break item in the neck, and the bone penetrates here, it will kill you. That's why sometimes neck break versus not just making sure being quadriplegic is so foramen magnum is there. The temporal bones, you have two of these, so it's because you have two ears. The temporal bone is right around where your ear is located. You can see here in yellow, either from the bottom picture or the side. But the bottom shows you both of them, 1 and 2. The side only shows you one. The feature I want you to know about this is, of course, we put your finger in your ear, that's the external auditory meatus. The other feature you should be aware of is that the temporal bone does have a sinus, but it's not close to the nasal cavity, it doesn't drain the nasal cavity, it doesn't have mucous membrane. It's a sinus bacterial mastoid process. It's of concern or of interest because inner ear infections, middle ear infections, can actually spread back to here and then from there to the brain. This is very rare. Number 4. The sphenoid bone, the keystone bone. The sphenoid you can see here is purple. Looks like it's divided. In fact, there's two of them. There's yellow division, but actually this goes all the way through. The yellow bone is on top of it, next to it. It's over here too, and it passes through the other side, so your temples, you put your finger over here, or over here on your greater wing than the sphenoid bone. Sometimes, at least the book calls it the keystone bone, because it passes through the skull, and a lot of facial and a lot of cranial bones connect to it. So that forms the keystone of a lot of the skull structure. That's part of the orbit of the eye. You can see here, but it's over here. It's on the other side too. I mentioned the temple. And there is a sinus here. So there is another nasal sinus. This one does drip into the nasal cavity. I can't really do a good job of drawing. It's deep in there. If it gets blocked up or infected, you feel it really deep behind your eyes. See that one or is the upper bone. The last bone of the cranium is one that we don't have a picture of. We can sort of see right here. It's clearly peeking out at the eye. I have another picture though. Right in the center of the nose here, underneath here, deep in the nose socket, you'll see a perpendicular plate and a couple of side structures, and those are part of the ethmoid bone too. So the ethmoid bone is a cranial bone. I call it the buried bone because it's so hidden. Unless you have a good cranial cavity picture, which this book does not have, you don't really see it. You see a little bit of it in the orbit of the eyes. Actually, quite a bit of it. They just don't have the names for it. So up here, and a little bit in the nose, and then you pop off here, you can see it down inside it because it's just a tiny piece that forms part of the wall of the cranial cavity, and that's why it gets classified as a cranial bone, part of the cranium. So it forms part of the orbit of the eye. It also forms part of the septum. And what is the septum? A septum is a divider, a division that divides the nasal cavity, in this case, into two cavities, the right and the left. So it goes right down the middle of your nose. There's a bony plate in there, and the ethmoid forms part of that. Additionally, the ethmoid has some sinuses, there's special sinuses called hair cells. There's a few of them. And like the sphenoid, it's very deep in the nasal cavity, drains in the nasal cavity, drains forms a lot of mucus. I want to make sure we have time for our last activity today. I'm assuming you already have the same paper. I meant to do this in the other order. There we go. So you can use the same piece of paper you used before. Alright, the answer to the answer form, use that same piece of paper, the back of it, or whatever. Don't need to bring out a new piece of paper. We're going to focus on writing the answers down, but if you're going to participate, which I hope you do, then you'll use your device, pick up the QR code on your camera, scan the code, or write it in on the website if you need to. go. Anybody else need the QR code? Once, twice, 3 times, and we're gone. Okay, remember to write down the answers even if you don't participate digitally. Link Okazaki fragments together. Ligase uses ATP, provides it with energy to take the Okazaki fragments on the lagging strand and link them together. So we have one complete strand. And that's its job. We've managed to find other things to do with it. Some DNA. Together. Make sure you're writing down the answers to these questions. They don't have to be correct answers, you can just be your answers. Be honest with yourself. Axons, folks. We got up our studying. I'm going to blame it on the lab exam. They're all worried about the lab exam. Anticodons are found on tRNA. Well, at least it's tied with the incorrect answer. Alright, so let's take heading back here. I don't know who Puppy is, though. The magical random. Dragonite? Type of genetically modified animal. Right, sterile mosquitoes. To my knowledge, they have not made slower growing salmon yet. Not a lot of them. According to scientific data, life began with that type of right, 3.5 billion years ago. The Earth kind of messed up. Earth's early atmosphere had little to no That's right, free oxygen. Little to no free oxygen. Oh, magical rhino wins out. Look and see the magical rhino. Okay. We'll never know. Alright, so if it's not a bone that forms part of the cranium, it has to be a facial bone. Now notice that before we had two cranial bones that were paired and the rest were single. Here, almost all the facial bones are paired with only a couple that were single. We start off with the two bones that sort of dominate the front of the face, the maxilla. You'll notice that the maxilla also continues quite a bit into your hard palate here. It meets in the sutural line right here. It has teeth. The upper arcade, the upper dental arcade attaches to your maxilla. You should be aware of that. There are a couple of sinuses on either side of the nasal cavity in the maxilla that drain into that nasal cavity. So that's an important feature of the maxilla. To the sides here, you have the zygomatic bones. So think of your zygomatic bones as your cheekbones. You can see they form part of the orbit of the eye. I'm showing a little bit of the sphenoid form back here. And then the nasal bone is, of course, part of the bridge of your nose. And the bridge of your nose only because this part is cartilage. I used to think this was elastic cartilage, but now that it's more, it's cartilage more like hyaline cartilage. That's why it's full, and it has a big empty hole here, because normally that's where we have cartilage, we don't have bone there, but there's bone that would be covered up. There are two of these nasal bones up here. No real feature to talk about. And then a tiny, tiny little bone back here is in the blue. The other picture too. Yeah, it's a little there, here, there. So it's on either side, and that leads into a little cavity called the nasolacrimal duct. So you got two of them. They're little small little rectangular bones on either side of the maxillae. Then the line nasolacrimal canal. This is a canal that goes to the nose. Lacrimal refers to the lacrimal apparatus which produces tears. So the lacrimal bones get their name from that. You actually produce tears on the superior side of your eye, and they drift down across the surface of your eye and lubricate your eye. And then they drain down into this canal, which leads to your nose. So you constantly drain tears into your nose. If you start crying, that's why your nose starts dripping, because you're literally crying tears directly into your nose. All in addition to all on your face. You're overwhelming your lacrimal system. The palatine bone is this bone back here, forms the last third of the hard palate. And named after the hard palate, though, there's actually quite a bit more of it that goes up into the behind the face. And so it's called a horizontal plate, and it's also a vertical plate. The inferior nasal plancha, whatever is translated. In the nasal cavity here, there are a number of what I call bony overhang, we call concha. They create shelves in your nose, which creates turbulence where you breathe in instead of the air going directly to your lungs. It gets bounced around in your nasal cavity and hits those. That helps to trap particles in your mucus so that it doesn't end up in your lungs. And most of them are associated with the ethmoid bone. That's that red is up there. That's the ethmoid bone up there. There's the perpendicular plane. These are the middle nasal concha. And then back down here, we have two bones. These green bones. These are bones into and of themselves called the inferior nasal concha. So they are facial bones because they're not forming part of the sinus cavity. They're just two little bones that project into the nasal cavity and create those two shelves. So they're relatively small, but they're bigger than the lacrimal bones. Our last two bones that are not paired, that are singular, include the vomer. There's a little bit of yellow down here forming the division. And then on the back, it's all vomer. And that's because the vomer is sort of wedge-shaped. It starts off narrow and then gets really wide in the back. This little wedge of bone helps to form the nasal septum. So that's what I want you to know is part of that divider that divides the nasal cavities into two chambers. And the mandible, there's so many things we can point out on the mandible. The mandible has a lot of features. But we're going to just focus on the fact that, of course, this is where your lower dental arcade is, this is where your teeth, where your lower jaw. This is the only bone that moves in the skull. Jaws were a big thing. There are lots of primitive animals, entire phylum that do not have jaws. The hyoid bones claim to pain as it does not articulate with any other bone. Now, if you put your hand right here where your jaw meets your neck, that's where the hyoid bone is. It is actually supporting a bunch of muscles that control your tongue and also for swallowing. When you swallow, this bone has to move up and down for swallowing purposes. So it's an important bone for supporting muscles that will in swallowing. So it has you for muscles of the tongue and other muscles that drive you to swallow. Looks like a little jaw in itself, a couple of little vampire pieces there. And it doesn't really touch any other bone. They investigate strangling victims or people who've been hanged, this bone is often broken. This is your vertebral column. This is what it's supposed to look. Runs at least the side. You got a nice cervical curvature at the top. You got a thoracic curvature here, and the lower back here, and the sacral curvature up here. A lot of pressure on this because of the fact that it has to bear all of their weight. If you get a side curve to this, so this, you would not see the side curve here because the side curve would be towards you or away from you. So if the vertebrae is curved in an S shape that's not this S shape, a sideways curvature, we call that scoliosis. And it's a fairly common problem. They put you in a back brace to try to fix it. But on the other hand, the thoracic bones here, if they curve too far this direction in the posterior direction, we call that kyphosis or hunchback. It's usually more pronounced up here, particularly when it's a developmental defect. The hunchback of Notre Dame is famous for its hunchback. If the lumbar vertebrae down here, if they curve too far forward, we call that lordosis or sway back. It's an anterior curvature too far forward. This is relatively common in women in the later stages of a pregnancy because of all the weight from the fetus up front here tends to pull on the lower back, and it leads to usually a temporary condition of lordosis that clears up after delivery. Okay, you have a total of 7 cervical vertebrae. cervical, neck. You've got 7 of those. You've got 12 thoracic. These are chest vertebrae. Of course, this is where you actually have attachment points for the ribs. Well, although you can see it, but the transverse processes and then the rest of the body meet, there are little dimples there where the ribs attach. So for that, and then you have 5 lumbar, this is lower back. We have 1 sacrum, but the sacrum is fused, made from 5 fused. I have a convenient term for sacrum. I think it was sort of in the region of the gluteal region, the buttocks. So if I talk about tailbones, that's the coccyx. This is often called the tailbone. It serves as an important attachment point for muscles that are involved in defecation or controlling muscles that are involved in controlling the gastrointestinal system. Exit point, let's put it that way. This is anywhere from 3 to 5 fused vertebrae. Alright, just say fused bones. Not really true vertebrae, like the smallest was a little tiny metal bone. Most people have 4, that's why the book report is 4. You got 5, a little bit of tail. So rather than types of vertebrae, I should probably call this vertebral structure. Vertebrae. So this is looking at the vertebrae from the top. This here up here is called the spinous process. You can feel these by rubbing your thumb right down the middle of your back, the little bumps back there. That's where all the spinous processes of vertebrae. The ones off to the side here are called the transverse process. This is actually called the vertebral foramen, and ultimately is where what forms the vertebral canal. That's where the spinal cord would be found for most of your vertebrae. Then if we put 2 vertebrae together, again, put a nice little vertebral disc in there again. This hole that they create when they come together is called the intervertebral foramen. And that's where we find the spinal nerves. So in there could be a nerve. And that would come out and go and control parts of your body. This is the intervertebral disc. And it is not perfect. It gets old, you get older, it wears out, you put too much stress on it, and it starts to bulge out on one side. This is made possible because the center is sort of like an inner gooey center called the nucleus pulpus, and it will push out the side, and this will start to push into the spinal nerve. That can lead to pain, numbness, paralysis. It's a very serious condition. It's called a herniated disc. It's herniating into that universal space. Another type of hernia is inguinal hernia, when the intestinal tract starts to push its way down into either the scrotal region of the male or the labia majora of the female, and it will cause it to outpouch, and that can be dangerous too because it can blood supply to part of the GI tract, and that can cause gangrene and sepsis and that. So not fun. By the way, herniations are not good. They're parts of our body bulge out where they're not supposed to. Any questions? For your ribs, we have 7 pairs of true ribs. True rib. You have a total of 5 pairs. Of false ribs and 2 pairs that are floating ribs. However, be aware that floating ribs are also false ribs. So this is a subcategory of the false ribs that floating ribs are. And we'll show you here. The first 7 pairs of ribs, all the way down, directly connect to the sternum. They have a cartilage that makes a direct connection to the sternum. Rib 8, 9, and 10 still have cartilaginous connections that will lead to the sternum, not this one, that will lead up to the cartilage of the 7th rib. So these all fuse together and make one connection to the 7th rib. So they're called false ribs for that reason. They do not have a direct connection to the sternum. False ribs, no direct connection. These last two, not only do they not have a direct connection to the sternum, they don't have any connection to the sternum. Rib 11, and back here is 12. They don't come around at all. They have no cartilage on their end. They only connect to the vertebrae, the 11th and 12th thoracic vertebrae. So they're called floating ribs because they're hanging out and they have no connection. They're floating. They're not strictly speaking true because you have connection to the vertebrae, but the lack of sternal connection makes them floating. The sternum itself is made up into 3 parts. The medubrium, which is where the clavicles connect. Unfortunately, they don't put the clavicles in this picture. They actually connect up here near the top with these what are called clavicular notches. The body of the vertebrae, the central part, and this little point down here, the xiphoid process. This is the last part of your body, typically, that will turn into bone. It's cartilage almost until you're in your 30s, or actually it is usually cartilage in your 30s, mid-30s that turns to bone. This is the part that they want you to be aware of when you're doing CPR. If you're pressing down on someone's chest, they don't want you hitting that and driving it into the liver because it will cause them to bleed out and die. They break it off. So this is an attachment point for the big diaphragm muscle. This is a breathing muscle, respiratory muscle. Body. And Ziploid process. And as I said, often, maybe you haven't heard in a while though, if you're having trouble studying and remembering, draw. Alright, that's a little too big. Help you to remember things or Shall we leave it there? Go to lunch?